Hello everyone, and welcome to episode 53, Remastered. Today, I'm continuing my series on plant physiology by exploring the subject of plant reproduction. Although there are numerous kinds of plants, from green algae to mosses and liverworts to great spruce trees to the flowering plants, I'm going to mostly focus on the reproductive physiology of this last group, of the flowering plants. And this is because angiosperms are the most common, the most useful, and the most harvested kind of plant. And in my opinion, they're just the most interesting and dynamic when it comes to plant reproduction. When I was talking about plants, way back in episode 33, I talked about the complex aspect of plant reproduction called the alternation of generations. I'll explore that concept again in this episode, and I'll be doing it right here at the very beginning, because, well, it's really fundamental to plant reproduction. Alright, so this alternation of generations means that the plant lineage has two different stages, or phases, that alternate as the lineage perpetuates itself, as the individual organisms within the lineage reproduce and go back and forth between these different stages. Now, really quick tangential note, in Series 3, I talked about genetics. I discussed that we humans are diploid organisms because our cells have two copies of our genome. We have 23 different chromosomes, and because we're diploid, we have two copies of our chromosomes, for a grand total of 46. Now, in plants, one of the two stages in this alternation of generations is a sporophyte. The sporophyte, much like us humans, is a diploid, which is to say that the sporophyte has two copies of its genome. It has two copies of each chromosome. Now, the other stage in the alternation of generations is the gametophyte, which is haploid. And a haploid, the prefix hap, means one, meaning it only has a single copy of each chromosome. The haploid gametophyte has a single copy of the genome in each of its cells. Alright, so moving on, the diploid sporophyte is so named because it produces spores, just like the gametophyte produces gametes. The sporophyte has reproductive structures called sporangia, and within the sporangia, the cells undergo meiosis to produce haploid spores, or spores that just have a single copy of the plant's DNA. So just like humans, who are diploid, with our gonadal tissue that undergoes meiosis to produce haploid gametes, like sperm and eggs, that's what these sporophytes do. These sporophytes undergo meiosis to produce haploid spores, or spores that just have a single copy of the plant's DNA. These haploid spores, produced by the diploid sporophyte, are then released into the air, where they fly away, floating through the air, and then eventually land somewhere, where they can germinate and then grow by undergoing mitosis. Without any kind of fertilization or input from any other plant, this haploid spore will grow into the haploid gametophyte. Now, the gametophyte can produce gametes simply by reproducing through mitosis. After all, gametes have a single copy of the genome. Gametes are haploid, and the gametophyte is already haploid, so it doesn't have to undergo any fancy-pants meiosis to produce haploid gametes. It's already haploid, it can just go through mitosis and pump out gametes that way. Except, there's a little bit more detail here. Some of these gametes are sperm analogs, while other gametes are egg analogs. When a sperm and an egg gamete meet and fertilize, they will create a zygote, which is a diploid sporophyte zygote. The cells in this zygote will divide by mitosis, growing into the sporophyte's body plan, which eventually grows specialized reproductive cells called sporangia, and these sporangia produce and release spores, and the alternation of generations begins again. The sporophyte and the gametophyte stages can be wildly different in size and shape when you compare different plants. In terms of size and shape and longevity and all these other different kinds of factors, the sporophyte and the gametophyte body form, these different stages, can be wildly different, not just from each other, but between different plants. There's just an immense amount of variety in these body plans and these reproductive stages. For example, in angiosperms, so in most flowering plants, The sporophyte is the main phase of the plant's life. It's the main body structure that will grow the largest and live the longest. If you're looking at a flowering plant with its flowers in full bloom, you're looking at the sporophyte stage of that species. The sporangia in the sporophyte is composed of two structures called an ovule and an anther. They both produce spores, but each produces a particular kind of spore. The anther produces microspores that will go on to become male gametophytes, while the ovule retains megaspores that will become the female gametophytes. So when you're looking at this large flowering plant, be it some kind of tree or a bush or something, you're looking at the sporophyte stage. But the sporophyte stage is so dominant in these plants' life cycles that the gametophyte is little more than a little growth that's kept on the sporophyte. These megaspores that are grown in the ovules, they'll develop into the gametophyte inside of the ovule. And then the anther that produces these microspores, as these microspores are being produced in the anther and then released, they are forming the small little gametophyte. That is the gametophyte. And so when these microspores are released, they effectively become pollen that intends to land on some other plant's flower and pollinate the megaspore that's being kept in its ovule. Except this isn't necessarily spores fertilizing spores. These are spores that have grown into gametophytes, and then the gametophytes are meeting and fertilizing each other. This fertilization of the microscopic haploid gametophytes will produce another diploid sporophyte in the form of a seed. In mosses, this priority is reversed. In mosses, the gametophyte stage is large and long-lived, and the sporophyte stage is tiny and dependent on the gametophyte. So in moss, the sporophyte is a thin stalk that grows out of the top of the female gametophyte, with a little sporangia on the end. The sporangia will release both male and female spores, which grow into male and female gametophytes. These gametophytes are the main structure of the moss, the main fuzzy tendrils of the plant. And at their very tip, the male gametophytes will grow sexual structures called antheridia, which will release a cloud of tiny sperm into the air. These tiny floating sperm released into the air will land on the female gametophytes' analogous structures that sit at their tips, which are the egg-containing archegonia. The released sperm will float through the air, or more commonly, they'll float in the water, like when it rains and soaks a tree, and all of that moss that's growing on the tree is now coated in a thin layer of water, and all of these sperm floating on the water can be deposited on the archegonia, where it will enter and fertilize the egg, and this fertilization will create a diploid zygote. This diploid zygote will then grow into a thin sporophyte stalk, which grows out of the female gametophyte and produces more spores, thus starting the cycle again. Except the whole time, the sporophyte doesn't leave the gametophyte. The moss's sporophyte is like a small growth coming out of the gametophyte. The flowering plants provide the most interesting angle on plant reproductive structures, and this is because flowers themselves are just incredible little organs. The flower is bright, fragrant, attractive to bees and humans alike. I talked about flowers briefly in episode 49, mostly about the gene expression that's involved in flower development. So today, I'm going to cover some of the same basic details, but talk more about the physiology and the function of the flower. So there are 4 distinct parts to the flower, which I already mentioned in episode 49. You have the sepals on the outside, the flower petals themselves, the stamen structures that release spores, and the innermost structure are the carpels that hold the ovaries. As you presumably know, flowers have a huge diversity in their appearances. Not every flower has every one of these parts, and the size and shape of the sepals, the size and shape of the flower petals, the scent, the fragrance, all of it varies a lot between species. The sepals are the outermost layer of the flower, and compared to the petals, the sepals are much more leaf-like. They're basically green leaves that wrap around the outside of the flower. All of the sepals in a flower are known as the flower's calyx. The purpose of the calyx, of all of these sepals, is largely protective. They're thicker and sturdier than the petal tissue, and when the flower is growing and getting ready to bloom, the sepals wrap around it and provide a degree of mechanical protection from bacterial and fungal pathogens, as well as herbivorous insects who would want to eat the flower's petal tissue. In many cases, the sepals continue to provide some kind of protective or structural role in the flower after it's bloomed. Now the next layer in is the flower petals themselves, which exist inside this calyx of sepals. As you almost certainly know, flower petals are the flashiest part of the flower. They have all of the beautiful color, they exude the scents that make the flower smell so good. And the reason these flower petals have all of these attractive qualities is because the flower is part of the plant's reproductive cycle. It's part of the plant's reproductive strategy. The angiosperms have evolved flowers as part of a symbiotic relationship to attract various flying species of pollinator, like bees or hummingbirds. The plants found that getting a little animal to carry your pollen from flower to flower was actually a really effective way to reproduce. Far more effective than just throwing your pollen out into the wind and hoping for the best. The colors of the flower petals, which are usually bright and vivid, are used to visually excite an animal, to get it to notice the plant and start paying attention to it. And the scents are used to attract the animal and to guide them towards it, to guide the animal towards the center of the flower where all of the pollen is. Some plants use nectar to incentivize pollinators, which is produced by little glands called nectaries at the base of the flower petals. The nectar that these nectaries produce is a sugary fluid that animals like hummingbirds and butterflies and all these other animals that they depend on for food. They drink the nectar through a long tube organ called a proboscis, which has actually created a kind of miniature evolutionary arms race between plant and pollinator. The nectar is sugar-rich, and so it takes a fair bit of sugar resources to make. It takes a fair bit of energy to make nectar. The plant benefits the most when this nectar can be used to entice multiple pollinators. But if one pollinator comes by and drinks all of the nectar, then all of that nectar just went into one animal, who may or may not have even carried off any pollen to help the plant reproduce. In this case, the plant just wasted all of that nectar with no benefit. It didn't get any return on its investment because its pollen didn't get spread around. Instead, the plant wants multiple pollinators coming and going, each of them getting a little bit of nectar, and each of them spreading around a little bit of the pollen, thus greatly increasing the potential reproductive capacity and the spread of the plant. If the plant can get more pollinators to come by, it has a better chance at reproducing, simply because it has more vectors going out, spreading its pollen around. So, to prevent one greedy hummingbird from taking all of the nectar, some flowers have evolved to make these deep, narrow basins, with the nectar at the very bottom. The farther away the nectar is, the farther it is to reach, and so the less that any individual pollinator will be able to take. But because the fluid-feeding pollinators, like hummingbirds and butterflies, depend on the nectar for food, they're fluid feeders and this nectar is virtually all that they eat, they will experience a very strong evolutionary pressure to grow longer proboscis, which only encourages the plants to grow deeper flowers, and in response, the animal is pressured to grow an even longer proboscis. This is the essence of the evolutionary arms race. Inside the ring-like corolla of flower petals are the two innermost layers, which are comprised of the flower's main reproductive structures. These are the stamen and the carpel. A flower might have multiple stamen, and each individual stamen is a tall, thin stalk, called a filament, which has a club-like anther at its tip. The anther is a bulbous little organ that produces the pollen, and when the anther opens up, or gets rubbed apart by some pollinator, like a bee that's walking around on the flower, that pollen within the anther will be released. The pollen can either catch the air and float into another plant's flower, or it can stick to the hairs or the feathers of a pollinator and get carried that way. The other reproductive structure is the carpel, and the carpel is composed of a stigma, a style, and an ovary. So the stigma is the upper portion of the carpel. It's like a little opening or a pore or a hole of some kind. And when a pollen grain comes and lands in the pore, when it lands in the stigma, it'll go down through a tube called a style. And this will guide the pollen to the ovary at the base of the carpel. The ovary contains little structures called ovules, which themselves contain the germ cells that undergo meiosis to create eggs. Now, the pollen are basically sperm gametes, as they're also created through meiosis. So pollination is thus plant fertilization. Pollination is a really interesting subject. Many species of angiosperm have flowers that possess both stamen and carpels, and each flower produces both pollen and eggs. Other angiosperms, called monoecious angiosperms, have both stamen and carpels, but individual flowers only express one structure or the other, not both. The monoecious angiosperms and the types of angiosperms that have both stamen and carpels in each flower, these are hermaphrodites, and they can self-pollinate themselves to create seeds. While self-pollination is really easy to do because all of the parts are there in one spot and ready to go, it does come with the penalty of lower genetic diversity. Then you have the angiosperm plants whose flowers only express one reproductive structure or the other across the entire plant. With these dioecious angiosperms, there can be male plants with flowers that produce stamen and female plants with flowers that produce carpels, but you won't see any kind of plant that has both reproductive structures. These dioecious angiosperms cannot self-pollinate, so they have to rely on pollinators or the wind to carry their pollen from plant to plant. Even though this technically makes reproduction harder for them because self-pollination is so easy, being forced to sexually reproduce offers a fitness benefit in that there's a healthy amount of allele mixing which preserves genetic diversity. So the monoecious plants and all of the other plants with hermaphrodite flowers have evolved a few mechanisms to avoid excessive self-fertilization, and thus avoid the risks and the problems that are associated with constant inbreeding or with low genetic diversity. In some cases, the cellular maturation of the male and the female gametes will take place at different times, so they can't self-pollinate, because only one kind of gamete is ready at any given time. So it's kind of like if the pollen is mature and ready to be released, but the eggs aren't mature yet, so they can't be fertilized in the first place. The plant can release all of its pollen, and it doesn't have to worry about accidentally self-fertilizing. Some other plants use molecular mechanisms, like some kind of signaling or identifying proteins on the gamete cell membrane. When the pollen and the egg from the same individual touch, the membrane proteins on one gamete will identify the other gamete as coming from the same individual. And because there's this recognition of the signal that says, we come from the same person, we come from the same individual, fertilization will be chemically rejected. Other plants will use a brute force method. They simply position their anthers and their stigma in such a way that they're really far apart. And so accidental or unintentional self-fertilization is pretty much impossible. It's simple, it's effective, and it doesn't require any fancy membrane proteins or meticulous timing of cell maturation. It's just a straight brute force method. Alright, so let's zoom in a little bit on the process of fertilization. How exactly does plant fertilization work? In most plants, fertilization is relatively simple, like it is in animals. Sperm meets egg. They chemically fuse, sperm dissolves and deposits its genetic material into the egg, the genetic material meets, it combines, that's fertilization, bam, you're done. But in angiosperms, this process is a little more complicated. I mean, in reality, all of this stuff is really, really complicated. So when I say that it's simple, that's relatively speaking. So in angiosperms, this already very complex process it's a little more complicated. It involves a second fertilization event. Now, before I explain that, before I get into double fertilization, I should really quickly talk about how sperm and eggs are formed, because how they form is very relevant to how plants reproduce. Inside of the anther, inside of these structures that make the pollen, there are these diploid cells called microsporocytes. And these microsporocytes undergo meiosis to produce haploid microspores. Each microspore undergoes mitosis to split in two, creating a tube cell and a generative cell. Both the tube cell and the generative cell exist within a single pollen grain, encased within a protective, watertight outer coating. Alright, so remember that. Each pollen grain has these two different types of cells within it. Now, inside of the ovule, there's a really big diploid cell called a megasporocyte, and the megasporocyte will divide by meiosis to produce 4 smaller megaspores. For some reason, 3 of these 4 megaspores will decay. They'll wither away into these tiny little non-functional blobs, and this leaves behind a single megaspore as the sole survivor. This single megaspore will expand to become an embryo sac, which divides by mitosis to produce 8 haploid nuclei. 3 of these nuclei will group up at the top of the embryo sac. 3 of them will group up at the bottom of the embryo sac, and the 2 remaining nuclei will remain in the center. And these 2 nuclei that sit in the center of the embryo are called the polar nuclei. Alright, so now the stage is set for double fertilization. You have this complex, weird setup, where each pollen grain is 2 cells packed into a single grain, and where each megaspore has 8 nuclei within it, but the nuclei are arranged in this weird, semi-symmetric pattern. So when a flower releases its pollen, the pollen grain will get carried by the wind or by some kind of pollinator to another flower, where it can land on the stigma of its carpel. Now the stigma, this little opening to the carpel, is kind of sticky, which makes it really easy for the pollen grain to be captured. Once held there, once bound on this sticky stigma, the pollen grain will absorb water, and much like a seed, it will germinate. Although a pollen grain is much smaller than a seed, so this germination process is similarly scaled down. Instead of a thick root coming out of the seed, the tube cell inside of the pollen grain will expand into a longer tube and snake its way down the stigma and down the style towards the ovary. The generative cell is carried down the pollen tube, where it then divides into 2 sperm cells, and these sperm cells flow down the pollen tube to the ovary. The sperm is deposited inside the ovary, where it makes contact with the egg and initiates fertilization to create a zygote. Alright, so I mentioned that these plants have a second fertilization event, and this is because there are 2 sperm that are going down the pollen tube. The first sperm will fertilize one of the nuclei on the end of the embryo sac. It'll fertilize one of the 3 nuclei in one of those groups on either end of the embryo. And the second sperm will go deeper in to fertilize the polar nuclei at the center. This single sperm fertilizing 2 nuclei creates a triploid structure called the endosperm. This endosperm becomes a nutrient-rich tissue that is used to feed the zygote in the seed during germination. The endosperm is kind of like a placenta in mammals. It's a tissue that's full of proteins and lipids, and the developing zygote will tap it for nutrients and chemical energy, and it helps the zygote grow at a time when it's obviously incapable of getting nutrients on its own. So when a seed forms, you have the zygote within the seed, and you also have the endosperm, which is like the placenta. The zygote and the endosperm will grow to fill the ovule, and the ovule's outer cover will harden so it turns into a seed coat. While the seed forms inside the ovule, the ovary that's outside the ovule will begin to change as well. The ovary tissue accumulates sugar, and it swells, growing into a fruit. I talked about seed formation in episode 49 on plant development, but because it's a part of plant reproduction, I'll go over the process of seed embryogenesis again here. If you recently listened to that episode, then some of this might sound familiar. So this freshly formed zygote will divide into 2 cells, an apical cell and a basal cell. The position of these 2 cells relative to each other will determine the vertical orientation of the plant's growth. It establishes a developmental axis to the embryo right away with this first cellular division. The basal cell will go on to divide into two cells. One becomes a cell in the root tip, and the other becomes the first cell in a chain of cells, like some kind of vegetative umbilical cord for the plant embryo. The apical cell will go on to divide and produce the vast majority of the cells in the plant. These apical cells will divide into three distinct cell lineages that create the three main tissues of the plant. You have the procambium, which becomes the plant's vascular tissue. There's the ground meristem, which becomes the plant's ground tissue or its cortex and pith. And then there's the protoderm, which develops into the epidermis layer of the mature plant. After a short period of cellular division and growth, distinct structures start to emerge. The lower part of the plant forms the radicle, which is the embryonic root. The middle part is the hypocotyl, which will form the stem. And then there's the upper part of the plant that forms the cotyledons, which become the first two leaves. Now, you might have heard of the terms monocot and eudicot. This refers to the number of cotyledons in the seed. Eudicot plants have two cotyledons, two of these embryonic leaves, and in most eudicot species, these generally absorb nutrients from the endosperm. Inside the seed, the cotyledons grow to wrap around the rest of the plant, creating a layer of green tissue between the endosperm and the hypocotyl. Because of their position as an interface between the seed coat and the embryo, the cotyledons tissue can absorb all of the nutrients out of the endosperm and become a kind of new storage organ or an intermediate organ that can efficiently shuttle nutrients through the embryo to feed its growth. In this way, the cotyledons and eudicots can act as the primary nutrient storage organ instead of the endosperm itself. The monocots have just one cotyledon, one embryonic leaf, and these simply tap the endosperm for nutrients instead of absorbing their nutrients entirely to take on the role of storage organ. When the embryo has fully matured and all of the tissues have differentiated and developed into vasculature, ground tissue, and epidermis, the seed is ready to go. But the seed can't just exist in a wet stasis forever. It would run out of energy and die and rot. Instead, the seed dries out, and its cells stop growing. For all intents and purposes, the seed is now in a dry stasis, and it waits until conditions are right before it germinates. Recall how I said that when the seed develops in the embryo sac, the ovary outside the embryo sac develops into a fruit? Well, this fruit is a really useful thing for the plant. And from an ecological perspective, fruit is really useful for the local community. For plants, the fruit is a soft but protective shell that encases the seeds, and the fruit serves as a mechanism for distributing the seeds outward. There's a lot of really creative ways that plants have adapted their fruits to serve these purposes. In many cases, the fruit simply dries out and falls off the tree, and then it rots and exposes the seeds. This is the simplest method, but it doesn't really get the seeds very far. It doesn't move the seeds very far away from their parent plant. Some plants have evolved fruits that exploit pressure to literally blow up and throw their seeds out and away from the parent plant. For example, the dwarf mistletoe fruit fills itself with sugar and thus pulls in water through osmosis, which will eventually result in a kind of controlled explosion, like a water balloon blowing up, which spews the seeds everywhere. The sandbox tree has fruit that dries out as it ages, and as the husk dries out, the husk will shrink. Eventually, the husk will shrink so much that the pressure of the seeds pushing against it from the inside will cause it to rupture, and the seeds, which have been held under increasing pressure for the last few weeks or months, will get blasted outwards and thrown tens of meters away. Now, all of these explosive techniques are interesting and pretty cool, but there's more effective ways for plants to disperse their seeds. Not just more effective, but they can disperse their seeds farther with these other methods. Some of these other methods include animals. Animals are a really popular and effective vector for seed dispersal. The animals are attracted to the sweet smell of the fruit, and they'll gobble it up off the branch, or in the case of primates, they'll pluck the fruit off the branch and eat it out of their hands. The animal will either haphazardly discard the seeds, or it will consume them and poop them out somewhere else. In this latter case, the pooped out seeds come out in a thick pile of fertilizer, which promotes the success of the plant after germination. So the poop is kind of like a little starter pack. It provides that nutrient-rich soil, that nutrient-rich growing medium for its roots to bite into and establish a foothold. Now, some animals will eat the fruit and poop out the seeds in a nice case of symbiosis, but other animals, like mice, might eat the fruit and the seeds. When the seeds get chewed up, the seeds are dead, and they're not going to be able to germinate and grow into a new plant. They've effectively been destroyed. To discourage animals like this from eating the seeds and thus ruining the plant's sugar investment, some plants have evolved defenses on their fruit structure, like the capsaicin in peppers that will burn an animal's mouth and scare it off. I should briefly mention that there are several types of fruit. The most common types are the simple fruits, like strawberries, coconuts, and radish, which develop from a single flower with a single carpel structure. Then there are aggregate fruits, like raspberry and blackberry, which are multiple fruits that are formed by a single flower, but that single flower has multiple carpel structures that can each produce fruit simultaneously. Then there are the weird ones, the multiple fruits, like the pineapple and the fig. These fruits form from multiple flowers. They create initial growths that meet and merge, literally fusing together to create a larger growth. This growth will then turn into the fruit. Some fruits can dry out as they mature, like a cashew or a walnut. Some fruits are solid and dense, like apples or pears. Other fruits are wet and fleshy, like tomatoes and peaches. But in every case, the fruit is derived from parent tissue, and it wraps around the seed, which is offspring tissue. I've always thought that was a cool little fact about fruit. If you're holding an apple or a tomato or some other kind of fruit in your hand, you're holding tissue that belongs to two different kinds of plants, both the parent plant and the offspring plant. The offspring plant is technically still just a seed inside the fruit, but it's still interesting nonetheless. Now, the last part in plant reproduction involves the seed detecting when conditions are right, and then germinating. But how does the seed do this? How does the dry, inactive seed sense its environment? Well, in warm, humid climates, especially tropical climates with no real seasonal differences and a perpetual growing season, seeds aren't adapted to be in any kind of dormancy. They just germinate right away. But in temperate and boreal climates with a summer and winter season, seeds have adapted the capacity to enter and exit a state of dormancy. I mentioned a hormone last episode called ABA, or abscisic acid, which plays a role in growth regulation by inhibiting cell division and germination. This growth hormone is largely responsible for stuff like storing nutrients, helping the seed go through its drying out process, and suppressing the seed's germination response. There are also hormones that I talked about called gibberellins, which do the opposite. These promote growth and encourage germination. For dormancy to be cancelled, or shut off, so that the gibberellins can be expressed and the seed can germinate, it has to undergo some kind of activation, or meet some kind of threshold. Some seeds need to be exposed to red light, which indicates that there's a relatively decent amount of sunlight available. Other seeds with thick seed coats need their coats to be scarified, or degraded and worn down so as to expose the internal tissue to sunlight and the air. Some seeds need a temperature cue to end dormancy and initiate germination, like the seeds of boreal plants that need to know when winter ends and when summer begins. Then there's plants that live in areas prone to wildfire. These wildfires can regularly clear out the underbrush, removing all of the local competition for water and creating a nutritious layer of plant ash over the soil. This is an opportune time for seeds to germinate, so the heat signal from the wildfire is an appropriate signal for germination. All of these germination conditions require water. Water is the necessary key ingredient. All seeds need it. Germinating when you aren't near water is stupid. It's a death sentence. So water is like the last signal that's necessary to begin germination. You have all of these other signals coming in, like temperature or the seed casing being degraded and exposing the embryo to air and sunlight, but water is the last signal that really activates this process. When water gets into the seed, it rapidly fills the cavity and diffuses through the plant tissue. The availability of water in the embryo tissue allows the seed to start consuming oxygen in cellular respiration and producing proteins for growth. After some time, water uptake will slow to a halt. The plant is as hydrated as it needs to be. At this point, the cells will really start to come alive. Proteins are synthesized, mitochondria fire up their ATP engines and begin multiplying, and the membrane expands as cells grow. By now, the cells are turgid with water pressure, and the embryo is ready to germinate. The root tip, the radicle, emerges first from the seed and reaches around to find soil. Finding soil, the root will dig in and grow, penetrating deeper into the soil and anchoring the plant to the ground. The stem will rise and lift the cotyledons up into the air. These new leaves will unfurl in the sunlight. Water is absorbed, carbon dioxide is inhaled, photosynthesis creates sugars, and the young plant begins its life. Alright, well that is about it for plant reproduction. That is more or less how the whole process goes. It's really involved and intricate, tailored by evolution to allow the plants to respond and time their growth in accordance with the growing seasons, so as to maximize their chances for survival and reproduction. Join me next week for the final episode in this series on plant physiology, where I'll be exploring the fundamental chemical process of photosynthesis, as it enables not just plant life, but virtually all life on the planet to live and thrive. If you like this episode, then hit that like button. And if you like all my content, then subscribe to my YouTube channel to get first access to all this stuff right when I post it. If you're listening to this episode on iTunes, then be sure to give us a review and leave a comment. That kind of stuff really helps spread the word and get the podcast out there. It helps bump the podcast up in iTunes charts, so more people can see it, more people can listen, more people can support the podcast, and in turn, I can keep making these kick-ass biological episodes. And as always, thanks for listening.